So it's my pleasure to introduce our first speaker this afternoon. It's Robert Ellis Hutchings from Dow Chemical. Robert is online this afternoon. Robert is a toxicologist at Dow Chemical and is an eminent expert in this area. And the title of his presentation is Microplastics in Human Health. What do we know and what are we missing? So Robert, over to you, please.
Yeah, thank you very much. Hey, I really want to appreciate and just thank Norm Kaminsky and the rest of the organizing committee for the invitation to be here today. My sincere apologies for not being able to be there in person. I'll tell you why a little bit later, but I'm at a microplastics workshop in Spain. But again, I didn't want to miss the opportunity to share with you some of the understanding that I have and others in the field in regards to human health and what we know about microplastics, and importantly, what are we missing? So the outline of my presentation today is going to be pretty basic. I want to talk to you about what microplastics are from a level setting perspective, talk about how the issue space is growing rapidly. I want to spend the bulk of the time talking about the state of the science as it relates to microplastics and human health. And then we'll talk about what we're missing. And I'll give you a little bit of an insight of what the industry is doing as an effort to help close some of the gaps that exist. So first, what are microplastics? Well, one thing to be aware is there's no universally agreed definition. What is agreed is that they are solid plastic particles less than or equal to 5 millimeters in length. Besides that, there's much debate in regards to what's included in the definition and what's not included. And there are a number of definitions. So for example, what is plastic? Is rubber considered plastic? Some would not consider that. What's solid? When you get down to smaller molecular weights, such as waxes, are those solid or not? What should be the lower size limit? This is often discussed. This is quite significant. How is insoluble defined? And should natural particles be excluded? In the EU REACH restriction on microplastics, that comes into play. So the important thing here is for science purposes, we can afford some flexibility, but when it comes to regulatory, definitions are critical and they matter quite substantially. So what are sources of microplastics? You can think of this in two different bins. One is primary microplastics and the other is secondary microplastics. So primary would be sources such that are purposely manufactured or produced. So things like personal care microbeads, pellets. So pellets that are produced, those get you into that 5 millimeter category, paint products, etc. And when we think of secondary microplastics, these really are the major sources. The ones that I have on the slide here are underlined as the major sources: debris from plastic breakdown, synthetic textiles and clothing, tire and road wear particles, and I have other examples here as well. This is not a well understood category, and so it's subject to change in terms of what we consider in secondary microplastics, primary microplastics, etc. So this is a very much an active area of our understanding in the issue space. What we do know is, and I'm sure you're well aware of the fact that microplastics is highly visible in the public domain. Not a day goes by that there isn't some sort of public announcement or news article about microplastics, whether it's in the environment or in a human exposure scenario. You hear about the issue a lot. Those concerns that have been expressed are translating into regulatory actions. And I just have a listing here. I'm not going to go through them for the purposes of time and wanting to get to the science. But the important point is that the concerns are expressed. It's ahead of the science. And so that's just the scenario in which we're dealing with. It doesn't negate the fact that there isn't exposure potential, but the answers to the health side of things, there are limited answers. So what I want to focus on for the bulk of the time today together is to talk to you about what we know about human health and microplastics and what we don't know. So first, we know that we are in this exponential growth phase of microplastics science publications. On the environmental side or overall, it started over a decade ago, but on the human health side, we're really only about 5 or 6 years in. And you can see that represented here by the yellow bars as it relates to humans. To put it into context a little bit, we have phthalates and BPA and PFAS on the 2021 axis. You can see that microplastics publications are 2 and 3x those other substances of concern. So it clearly is taking a prominent role in terms of scientific research. Where is that research being conducted? So 40% approximately of the publications are coming out of China. The other area that it's really taking a lead in terms of publications is the European Union, particularly as it relates to funding of microplastic science. There's many other areas in which good microplastics scientific efforts are being conducted, and I listed some of those down below. But really leading the publication space is China and the European Union. One note about Europe in particular that I think is important to share, the way they do their research projects is in clusters and consortia. So on the left is a human health cluster of different groups that are covering various aspects of human health, whether it's looking at farm exposures, looking at air exposures, focus more on immunotoxicology, etc. So there's often overlap with some of these cluster projects, and particularly with COVID, there were delays in regards to these clusters getting together and making sure they weren't duplicating efforts and being able to advance projects. On the right-hand side, there's the Momentum Project, which is a very large human health project that has actually leading the way in terms of these European cluster approaches. And on the bottom is this CoreNet project called Microplastics at Food. I bring it up here particularly because of Chris's focus on food, but it's also one in which impacts human health, and it highlights the challenges that exist in this field, namely methods development, considerations of analytical challenges, quality control. These are all being tackled by this Microplastics at Food project. It's a 2-year pilot program, and they're just now looking at funding for the next phase called Microplastic at Complex Food, looking at more complex matrices. So I'll come back to this project in a little bit, but this gives you a little bit of an idea of what's going on in Europe, which is a hotbed of activity for microplastics and human health science. Really what I want to get through today is talking about what do we know about the impact of microplastics on human health. In order to address this, we need to understand 3 aspects that then get incorporated into risk assessments. First is what's our knowledge about external exposures in the air, food, and drinks. Once you get the external exposure, does that get taken up? Does it get absorbed, distributed? How is it eliminated in the human or model systems? Once it gets internalized or point of contact, is there actually a hazard or an effect? And once you take all of that information in together, you can make a risk conclusion for any effects that have been identified. So that's pretty basic toxicology, but it gets more complicated as we apply it to particles and microplastics. So first, I'm going to spend just a little bit of time on inhalation. Most of the talk is going to be more on the ingestion side, but inhalation, just like other particle types, we know that particle size is a critical determinant. So PM 2.5 and PM 10 have been known for some time in terms of cutoffs for knowledge about effects of particles. If we transition to focus on microplastics, if we think about indoor and outdoor exposures, what we know is that indoor is likely a combination of the degradation and fragmentation of textiles as a key contributor. On the outdoor side, tire and road wear particles are showing themselves to be a key source of microplastics exposure. Another aspect is that of uncertainty here is that the respirable fraction, so 4 microns and below, it can't be estimated reliably at this point because the methods don't exist. The collection approaches for PM 2.5 and 10 don't cover well for some of the microplastics. So this lack of data and specifically the contribution of microplastics to PM 2.5 and PM 10 is an area of uncertainty that really prevents reliable estimates. So transitioning over to food and drinks from the ingestion side, what do we know about external exposure? Well, we know that there's a number of reports that have come out purporting exposure to people or babies through the food and drinks. Many of you are probably aware of the tea bag publications that have come out. The big question here is we don't understand quality control, quality assurance. There's a large, we know that there's particles everywhere. So how do you account for that when you think about methods? Particularly when you're developing methods for microplastics, there's a lot of potential for contamination of the samples. And again, I go back to this microplastics at food project that really is narrowing in specifically for food and food packaging some of the concerns around this space and trying to advance the science to improve the accuracy of the detection and the conclusions that are being made from some of these studies. So after external exposure, what do we know about internal exposure? So again, there's a number of publications that have come out in terms of people. So feces, there's been publications about human blood and lungs. These are relatively recent, but again, the same questions apply as it relates to what type of conclusions can we make? Well, I think we can conclude that there is the potential for exposure, but beyond that, it's really difficult to make conclusions based upon the quality of the methods that exist to date. So there will be more need to come in this area. So let's talk a little bit about uptake as it relates to the ingestion route. We know that the small intestine is a critical organ for uptake of particles, but specifically microplastics. The European Food Safety Agency indicated that particles of less than 1.5 microns can penetrate the GI tract. Again, particle size is critical. One thing that's known also from the available data is that if you have a particle in the 1 to 2 micron size, the uptake is about 0.3 or 0.1 to 3%. So we're talking about low uptake potential, and most of that would get excreted in the feces. As the particle size decreases, the propensity for uptake increases inversely. We know that there's modulating factors besides the primary determinant of size, such as surface properties, shape, diet and lifestyle, and pregnancy and lactation. Particularly on the latter, we know that blood volume increases during pregnancy. Some membranes become more permeable, and so that could increase the chance to some extent of microplastic uptake in the gut. One thing to point out is that we're starting to understand a little bit more about steady state. So there's some good work out of Bart Coleman's lab. Here is Nur et al. Looking at modeling of steady state microplastics in the GI tract. Again, using that 0.3 uptake value, they calculate a median abundance of 300 to 500 particles. It's expected to be higher than that. We don't know how much higher than that, but it's drastically different from what you may have heard about the 1 credit card a week reports that have come out. That is the 99th percentile of intake for that Nur et al. Modeling work. So that is most likely not representative of what the normal uptake would be. We don't know where it is, so there's a lot of uncertainty still in this space. One thing I wanted to point out is that there's a lot of overlap between microplastic particles and general particles in the environment, or natural particles. So on the left-hand side, you can see contour plot, and if you look at the purple, that's the microplastics. On the x-axis is size, on the y-axis is lifespan. And you can see a lot of overlap between natural particles and microplastics. Not entirely overlapping, but certainly a significant amount. And so it just highlights the challenge that exists in this space about microplastics and differentiating those from natural particles. On the right-hand side is similar. This is overlap in fibers. So between blue microplastic fibers and natural fibers, you can see they're almost directly overlapping. So This is one aspect of the challenge that exists in this field. On the bottom is abundance. Again, microplastics is that first column, and you can see that there's a lot of overlap between microplastic concentrations in regards to our knowledge to date and other natural particles. So there is overlap. We know that we're exposed to a variety of particle types. Our job here is to try to discern the effect or the exposure of microplastics relative to these other particle types. All right, so moving past uptake, let's talk about the hazard and eventually then the risk assessment side of things. What do we know about microplastics and effects? Do they cause adverse effects? There's a whole host of publications that come out, have come out and continue to come out, and they show anything from no effects to effects on specific organs, reproductive, even microbiome. I know we're going to hear a talk later in the session on the gut microflora. So there's a variety of effects that have been purported. One of the challenges that we've been involved with is how do you screen those different reports for their quality, their reliability, and their relevance? And so one approach that we've taken through a tripartite group is to develop a screening tool for microplastics because they're different from chemical substances. So we need to think about them perhaps in some of the same ways and some different ways than we would for chemicals. And so I was part of a project where we looked at developing a tool that took into consideration particle characteristics, study design, relevant for risk assessment, to try to have a tool to evaluate the existing science, but also to encourage improvements in the quality of the science. It was a tier 1 screen. It was not designed to be the end-all be-all for assessment of specific science. But it was taken up by the WHO in their recent drinking water and food and inhalation assessment. So they evaluated 109 studies as part of that, and I'll give you a little bit more later. It doesn't end with a tier 1 screen for reliability and relevance. You need to take that to an expert evaluation approach. So some of you may be familiar with the mode of action human relevance framework. An approach like that needs to be considered also for microplastics before one can reliably make a conclusion about risk. That is very important. So I want to transition to talk a little bit about what have regulatory authorities assessed as it relates to microplastics in human health and what conclusions have been made. So we see as early as 2016, the European Food Safety Agency has evaluated microplastics, the German Risk Assessment Group, BFR, as well as the French Risk Assessment Group, ANSYS, Canada, and more recently, the California group, SQUIRP, assessed human health and microplastics. I want to focus in a little bit on the most recent WHO report that just came out. And I want to use that as an example to talk about the key messages and what the needs are. I want to just lay it out for you because it's really critical on what they're saying. So first, exposure. Their conclusion is that human exposure to nano and microplastics is ubiquitous and occurs via all exposure routes. But we need data on all of these routes because it's so limited to be able to better understand what that exposure is. As it relates to epidemiology or occupational exposure, we know that there's evidence of nano and microplastics could affect the GI tract and other organs. Really, it's focused on the data that's available is focused on the lung and occupational. These other areas have limited information. And because of that, we need to have better estimates and information for the general population. As it relates to dosimetry, we know that there are mechanisms for natural particles that will limit tissue exposure, but we need to understand how microplastics differ, if any way, from that. We know, as I mentioned earlier, that the size is critical, and as the size gets smaller, the probability for uptake increases. We have insufficient information in this area, including the potential for them to cross biological barriers after that exposure occurs. And it's not possible currently to move from data collected in vitro to in vivo. That in vitro to in vivo dosimetry or extrapolation is not possible yet for microplastics. Also, key messages, effects. We know that most of the data that exists is on polystyrene beads. These are spherical feeds that have been produced primarily for analytical purposes, not for tox testing. And those have limited to very, very limited relevance to what would be, in this case, a food packaging type of microplastic that's been formed. We also know that we have many needs in terms of size, shape, polymer composition, and other factors that are related to what's actually in the environment and relevant. And we also need to have a better understanding of particles, how microplastics relate to other type of particles that already we have exposure to. And particularly mode of action type of work should focus on oxidative stress and inflammatory responses. And besides that, the quality control aspect is, again, very, very critical in this space. One aspect that I haven't brought up is nano and microplastics as a pathogen vector. You may have seen publications or news articles in this space. It's unclear, the WHO indicated that it's unclear if the microplastics associated microbial communities are different than for other particle types. And we need research to understand if there could be an infection associated with that and if humans could have exposure. So those are the key messages from the WHO. In regards to the basic needs of science, they came up with a number of needs. First, standard methods. We need standard methods that are robust, they're quality assured in all of these different exposure routes. There's a need for suitable reference materials that are representative of what's in the environment. When it comes to particle characterization, we need, again, good quality controlled studies in the environment to know and characterize what the actual distribution is, so that those can then be used in studies for human health. And they can also be, we can prepare reference materials that can be used in testing. So what do we know about sources? There's still a need to understand what the true sources are. We know, as I mentioned earlier, that there's tire and road wear particles, there's textile, there's degraded and fragmented plastics. But what we don't know is what source is most prevalent, where exactly are larger contributors. So there's still a need in that space. Uptake and fate of inhaled and ingested nano and microplastics. There's limited information on the kinetics. We need more information on the absorption, distribution, and elimination. We need information, particularly from the food matrix, on bioavailability of ingested particles and how that food matrix affects absorption and elimination. And the WHO also commented on needs in toxicology, particularly the need for quality assured experiments that would be suitable for risk assessments and that they would be using materials that are adequately characterized and inclusive of, again, environmentally relevant materials. I referenced standardized study designs such as good laboratory practice or good scientific practice, use of regulatory test guidelines. All of this together will increase the applicability of this work ultimately to a human health risk assessment. So just in these last few minutes, I want to talk a little bit about what the industry is doing to try to close some of these gaps. I'm currently at an International Council of Chemical Associations meeting, ICCA, to talk about this type of work. It's composed of regional associations that are all contributing various aspects of microplastic science to ultimately get to a risk assessment. And you can see that they're focused in analytical, exposure, and hazard aspects. The goal here is very similar to what I just walked through with the WHO work, is to get standardized methods and high quality information. And specifically, when it comes to human health, on the left-hand side for hazard, there's projects under Cefic in Europe looking at inhalation. PlasticEurope is sponsoring projects looking at the ingestion pathway. And you see similar approaches on the exposure side with uptake and distribution work being funded by PlasticEurope and composition of indoor and outdoor air funded by American Chemistry Council. One slide here on the human health as it relates to PlasticEurope. I'm not going to go through all this, but this project is just initiating. It really focuses on the ingestion side of things. covering everything from production of materials from commercial grade products all the way through the risk assessment. And it includes a tiered-based approach for in vitro, ex vivo, and in vivo testing, covering both the kinetic side and the toxicodynamic side. And you can find more information at this website that I've listed here. Ultimately, what do we need? We need a human health risk assessment, and particularly one that we're developing or starting to develop is to try to take into consideration both the traditional approach to risk assessment as well as the next generation approach to try to incorporate all the in vitro and ex vivo data towards a hybrid risk assessment approach that can be utilized ultimately for the needs that exist in this field. This MARI meeting that I'm at right now, this group is trying to advance collaboration and information exchange. You can see this here. The group has presented in a variety of venues. So on 2022, you can see the various scientific conferences that MARI has tried to disseminate information about what's going on. Next year, we've got the SOT meeting, CTAC, and there'll be more to come in ways in which you and others can understand what the industry is trying to contribute in this space and opportunities to interact. So in conclusion, I just want to remind everyone that this issue space of microplastics is a rapidly growing one. There haven't been conclusions to date, really because the representative materials are not being used in the studies. The quality of the studies are limited in terms of their suitability based on their low overall quality. There are a lot of efforts underway to close these gaps, not only by industry, but of course by academics and government regulators. The goals here are to use relevant test materials with real-world exposures and to generate high-quality studies using fully characterized materials, robust study designs that are suitable for these risk assessments. Industry is important in this space, but there's so much need for various stakeholders. It really requires all hands on deck. Thank you very much, and I'll take any questions you might have.
Robert, thank you for a fascinating presentation. Any questions in the audience to start out? Norm?
So Rob, thank you very much for that presentation. I had a question for you. I've been starting to go through the immunotoxicology literature, and I have to say I'm struck by what I'm seeing in these studies where they're all in vitro and they're putting somewhere in the order of 1 million particles onto cells and then claiming oxidative stress and release of inflammatory markers. And I've just focused on the immune system. Is this kind of what's happening throughout other organ systems? And I mean, a lot of the work is really, really questionable.
It is. The challenge, Norb, in this space is that, so for example, if we take a chemical, right, we have a good understanding of what we would want to test in terms of a solubility limit, right? We might use that, or we might use a well understood limit for just what's relevant, maybe 1 milligram per liter, right? But in this space, you don't have those standards. And so because the science is in its infancy, you just have folks that, and we don't know what the true exposures are in terms of numbers. And so people just put it in without thinking about it, right? So you're right. I mean, there are often studies that are looking at 10,000, 100,000 particles. And if you could look at the cells, you would just see a coating of particles completely swamping the cells. So, the question is, how is that relevant? And it's an appropriate question. So this is the type of dialogue that we need. We need to ask the challenging questions of, well, what would be an appropriate in vitro exposure? We need to ask the same question about in vivo studies when we do those, because we have limit doses for regulatory studies, but I would advocate not using that because, again, it's not going to be relevant. We need to find what the balance is through dialogue scientifically.
Okay, we have another question in the audience here.
Hey, Rob, it's Christy Says. It's nice to see you.
Oh, glad you're there.
Question is about one of those WHO key messages, the last one, the fifth one, about microplastics serving as some sort of vehicle for pathogens to maybe latch onto, and then the exposure of the pathogen is through the microplastic particle. And certainly there's not enough scientific data out there to suggest that is the case. But I would say that even if the WHO doesn't think that microplastics are any different than other types of particles that can do that, I still think it's significant because of the amount. Well, it's arguable if the amount of microplastics are greater or less than now versus, in the past, which also needs to be understood as well. But I would say it doesn't really matter, I would say, if the microplastics are different or the same as other types of naturally occurring particles or anthropogenic particles that can also bring pathogens across barriers and things like this. Yeah. And it's not only pathogens, but it's also other pollutants, contaminations in drinking waters or things like this. I haven't published the data yet, but my grad student and I, we've done some isotherms with paraben, and we do paraben in water and monitor its concentration, HPLC, over time. And when we put the microplastic particles in there, the paraben concentration in the water gets lower and lower, almost, immediately goes away. So, but that doesn't mean that the paraben isn't in the system. It's just absorbed onto the surface of the microplastics. And then that causes my secondary concern of, okay, now it's a complex exposure of paraben and microplastics whenever the exposure does happen.
Yeah. Yeah, I think there's 2 points you raise. One is that I think it's appropriate that the WHO included the pathogen vector as an important research area, right? So I think it's important that they called that out, and the fact that there's needs in that space. In regards to the other vector, right, of chemical pollutants, right? We know that plastics are used as semi-permeable membrane devices for environmental monitoring. So we know that plastics can act as a sink to sorb chemicals. I think the key question is, under the environmentally relevant conditions, can plastics be a significant contributor to everything else that's there? The science that I have seen to date indicates that it has minimal contribution, but obviously it's a very active science area. I think if you have a pure system, like you were talking about, let's say if you have just a chemical of concern in water and you put in microplastics, you're going to get a dynamic equilibrium that's trying to be established. I guess I would go back and say, well, how does that relate to the true environment and all the various contributors that would be present in that space? So I think it's an area that still needs active research, but it's complicated.
Okay, any other questions from the audience? We have one back here. Norb, any online?
I've got a hand raised, but I can't tell where it's coming from.
Okay, so are you gonna, have you found it or? All right, so let me get this side of the room and then I'll come over there, okay?
Hi, Robert. Very nice presentation. My question is like, how does the exposure of microplastics affect the integrity of intestinal tight junctions? Because the intestine is essentially, what's sealing it from the microbiome and other pathogens is the enterocytes. These are the single layer of cells that form very strong tight junctions. And we know that in certain diseases, such as inflammatory bowel diseases, these tight junctions get disrupted and can lead to various kind of, health consequences. So does the exposure to microplastics make those tight junctions more permeable or not? Is there any studies on that? Thank you.
I haven't seen studies that indicate that microplastics make them more permeable. I could be wrong. I could be missing studies. I have seen studies that discuss if you already have a disrupted GI tract, so let's say inflammatory bowel disease, could you get an increase in microplastics transport or uptake across the membrane? That's where most of the research I've seen is generating data. But I think we'll hear a presentation later that could probably do a much better job of answering that question than I could do. But it is a very active area.
Hello, Rob. This is Baoshan. I'm the last speaker of this.
Yeah.
I do have a question regarding nanoplastics. You mentioned quite a bit about the microplastics.
Yeah.
Currently, we don't have any standard or capability to detect, to isolate, detect nanoplastics. When you see we may overestimate the intake, I would argue differently because simply we are unable to detect nanoplastics. We may underestimate the intake or exposure. Would you take?
Yeah, I totally agree. Just frankly, the methods aren't in place to be able to assess nanoplastics. We're barely able to get down below 20 micron in terms of microplastics. So we don't know, right? We don't know how far plastics degrade, right? The presumption is that they will continue to grade below microplastics into nanoplastics. We don't know what the current situation in terms of nanoplastics in the environment. So these are open questions. And as I mentioned in my talk, as the size gets smaller, and size being a predominant determinant of uptake, the expectation is that you would get more taken up and to have the potential for biological interactions in the smaller space. We just don't, A, have the methods, and B, some hazard data could be generated using material, but we can't put that into context currently.
I have one question for you. You mentioned at the beginning of your presentation something to the effect that, if I got this right, that the regulators are out ahead of the science, which it's obvious why that might be the case. Do you see anything on the horizon? Are there any significant regulatory actions in this area that you foresee in the near future that would be of interest to folks who are either producing food or consumer products?
I think the main area of regulatory activity is focused on understanding the potential for microplastics to be entering into the environment. So pellets, so in the plastics industry through Europe, one of the activities is increasing our accounting for if there's any sort of pellet loss into the environment. So that's an area that is going to increase. The other area that will increase is going to be monitoring activities. So in California, they have a legal a law in place to start doing water monitoring. I think other states are looking at this as well. So I think the major area that we're going to get activity is going to be looking at a better understanding of what type of monitoring of microplastics could be in various matrices, starting with water. So I think that's the major area in addition to reporting